Hello, everyone, and welcome back to Chapter 6 of Flight Physiology and Human Factors. We are going to focus today on vision, which you can surely imagine is a very important part of safely operating an airplane. So, our chapter objectives for this are going to include understanding the anatomy and physiology of the eye, learning how light is refracted and absorbed in the retina, and how we convert that light into the electrical signals that are sent from the eye through the optic nerve and into the brain, and how we process that incoming visual information to actually produce images that we can see and perceive to make decisions while we are flying the airplane. We also want to understand the limitations of human vision. It's not a perfect process, and we can be fooled by all sorts of optical illusions, which we will learn about later. So we definitely want to understand kind of the inherent limitations of our visual process and how defects in how our vision is set up and how optical illusions can create problems for us and how we interpret visual cues during flight. And then finally, we want to talk about strategies for overcoming those visual limitations. And particularly, we want to talk about avoiding midair collisions. And we'll also spend some time talking about the unique challenges presented by flying at night. Okay, so the first thing we need to talk about is light itself. So light is more technically called electromagnetic radiation. So light, as it is, exists simultaneously as two different things. You can think of light as a wave or a particle. Okay, so it might be more helpful, kind of how you've always thought about light, to think of it as a particle. But for our purposes, it's going to be most useful to think of light as a wave. So that's how we're going to talk about it for the next couple of slides. So the reason why we want to focus on the wave properties of light is because it is those wave properties that cause light to be split into several different types of light. So we have light of varying wavelengths and energies. So if you picture a wave of light that looks like this, like you see in green, we can use several different measurements to describe that light and categorize it into the type of light that we're talking about. The first thing we want to talk about is called the amplitude, which is essentially how tall the little crests and peaks of the waves are. So the reason why this is important is because the amplitude of your wave or of your light reflects how intense or bright the light is. So we'll spend some time later in this chapter talking about how the brightness of light, whether it's dim or very bright, affects how your eyes adjust to darkness. And there will be times where you're going to be flying at night and in the dark. So the brightness of the light that you expose yourself to is actually going to be very important. So for that, we are talking about the amplitude of the light. So the wavelength of the light is actually going to be what tells us what type of light that we are dealing with. So the wavelength just refers to the actual distance from peak to peak or trough to trough of the light itself. And this will be standardized for each particular kind of light that we are dealing with. So specifically, this isn't super important for our purposes in this class, but the wavelength will also tell you how much energy is in the light. And this is an inversely proportional relationship. The shorter the wavelength, the more energy that light has. So what you're seeing in this diagram from your textbook is the entire spectrum of electromagnetic radiation. So we're not so much going to focus on the frequency to the far left, although of course frequencies are very important for radio communication in flight. What we want to focus on is the wavelength over here. So at the very top of this diagram, we have the electromagnetic radiation or light with the absolute lowest, shortest wavelength. So you have gamma rays, which are 0.1 angstroms. So that is 1 times 10 to the negative 11th meters. And then we're working on the metric system here. So in general, we're going up by an order of magnitude of 10 every time I move my mouse down. So 0.1 angstroms, that's 1 times 10 to the minus 11. 1 angstrom, that's 1 times 10 to the minus 10. A nanometer is 1 times 10 to the minus 9th. 1/8, and so on and so forth. And then at the very bottom of the diagram, you have your very long wavelength radiation. So 1,000 meters, 100 meters, 10 meters, 1 meter, and so on and so forth. So like I said, it is the wavelength that determines what type of light you are dealing with. So the very long wavelength radiation, you have radio waves. You have your long wave radio waves. You have radio and TV waves. You have microwaves, radar waves, infrared radiation. And then in your short wavelength, you have gamma rays, X-rays, and ultraviolet or UV rays. So what we in particular care about is this very narrow portion of the spectrum here, which is called the visible light spectrum. The visible light spectrum, or the light that we actually are able to see with our eyes, actually makes up a very small fraction of the electromagnetic radiation spectrum, only in the range between 400 nanometers and 700 nanometers. And the particular wavelength will actually determine what color you are looking at. So the shorter wavelengths are going to be your blues and purples. The longer wavelengths are going to be your oranges and reds. And we'll talk about how that actually works in terms of what determines what color something is in a little bit. So again, the radiation with the shorter wavelengths are going to be very dangerous and very harmful. So UV radiation, which causes sunburns, X-rays, gamma rays, these are all harmful because they have such short wavelengths. That means they have a lot of energy in them. If you get exposed to this kind of radiation, it can damage your living tissue. But the radiation with the longer wavelengths like IR, microwaves, and radio waves, these are harmless, and of course they're very useful for communication technologies. Now in particular, radio waves are all around us all the time. Your body right now, as I am speaking and as you're listening to this, the cells of your body are constantly being bombarded with radio waves. There are radio stations all over the country. Radio waves are just constantly all around us. But because radio waves have such long wavelengths, they cause absolutely no threat to us whatsoever. So let's talk about how visible light works in terms of what we see and what colors we are seeing. So the reason we can actually see objects that are around us is due to the way that visible light reflects off of those objects and how that reflected light interacts with the light-sensitive cells in the back of our eyes. So if you look at this picture here, we are looking at an apple, which of course is red, and then the leaves on the stem of the apple, which are green. So the light that we use to see ultimately comes from the sun, unless you're talking about artificially generated light. But if you go outside and you look at an apple on the ground, the sun is providing you with what we will call white light. White light is visible light that consists of a mixture of all wavelengths in the visible spectrum. So the reason why this light is represented as rainbows is because it is meant that that light contains every wavelength from 400 to 700, okay? So the sun is constantly bombarding us with all wavelengths in the visible light spectrum. We also get UV radiation. That's how you get sunburns. A lot of the harmful wavelengths get diminished in Earth's atmosphere. Okay, so when this light strikes the skin of the apple, which is red, or strikes the skin of the leaf, which is green, the question becomes, why does the apple appear red? Why do those leaves appear green? Well, what is happening here is each object that has a particular color contains pigments that will absorb particular wavelengths while reflecting others. So what you're seeing here is that when that white light strikes the skin of the apple, the pigment that is in the apple is going to absorb the purple and the blue and the green and the yellow and the orange, but it's actually going to reflect the red. So there's something about that long wavelength of the red variety that is not absorbed by the pigment in the apple and instead gets reflected. The same thing applies to the green skin of the leaf. It's going to absorb every wavelength of the visible light spectrum except the green. That gets reflected. Well, what happens to that reflected light? Well, that reflected light gets reflected in the direction of our eyeballs, And that light will pass through the front of our eye to the retinal cells in the back of our eye. And those particular wavelengths, and keep in mind, this is no longer a mixture of wavelengths. These are very particular red and green wavelengths that are now interacting with those cells in the back of the eye. And that is how we perceive those objects as being red or green. It's because those very particular wavelengths that correspond to very particular colors are exciting those cells in the back of our eye. Now, if we were looking at an object that reflected all of these colors towards our eye simultaneously, meaning that we're getting simultaneously the purples, the blues, the greens, the blues, and all that, then the object would appear white to us because it is a mixture of all wavelengths. If none of these wavelengths got reflected and all of them got absorbed, the object would appear black to us because black is the absence of light. So if all of the light is getting absorbed by an object and you are seeing no light reflecting off of it, then that object will appear black to you. So the thing you want to remember as we move forward is that we can only see wavelengths of light that fall within that visible light range. We can't see light that is less than 400 nanometers, and we can't see light that is greater than 700 nanometers. So we're going to learn later on that this is because of limitations in the photoactive pigments that are in our light-sensitive photoreceptor cells. So we'll get to talk about that a little bit later. So let's talk about the very basics of how this visual process actually works. So let's say that you see the light that comes from something like a flashlight. So that visible light is going to pass through the front of your eye, and we'll talk about the structures in the front of your eye later on. But that light is going to stimulate the retina, which contains the light-sensing photoreceptor cells, the rods and the cones that we'll talk about later. And this is going to convert that light energy into internal chemical energy. So these photoreceptor cells, which contain these photoactive pigments that we'll talk about later, they're going to convert the light energy in those photons of light, convert it to internal chemical energy, and then use that chemical energy to send an electrical signal through one of your cranial nerves, which is called the optic nerve. Each one of your eyeballs has an optic nerve, so you have two optic nerves. This is the pathway that takes that information about what you are seeing from the cells in the retina of your eye towards the brain, where the brain is going to process that visual information. This information makes a little bit of a pit stop at a particular structure in the middle of your brain called the thalamus. The thalamus is just a sensory relay center. It receives incoming information about what you're seeing, what you're hearing, what you're touching, what you're tasting, and it distributes that information to the appropriate processing centers in different parts of the brain. For visual information, the thalamus is going to send this information to the visual cortex, which is located in the very back of the brain in a part of the cerebrum called the occipital lobe. So occipital sounds a little bit like optic, so that makes it kind of easy to remember that it deals with vision. So as far as how the energy is treated here, again, we are converting the light energy from these photons of light that enter the eyeball into chemical energy in the photoreceptor cells, and then finally into the electrical energy that runs through the nerve pathways from the eye to the brain. Okay, so now let's talk about the actual anatomy of the eyeball itself. And don't be discouraged by all of the labels here. We're not going to talk about every single structure here, so don't worry too much about that. But for now, let's split the eyeball into two parts. We have a front or anterior segment, so that's everything to the left over here, and then a back or posterior segment. That's everything over to the right over here. So the anterior and posterior segments have very particular functions that we're going to try to keep track of. The anterior segment, again, that's everything over to the left over here, the anterior segment is going to focus on bending, refracting, and focusing incoming light and pointing it towards the retina back here. So the anterior segment does not contain any sensory cells for detecting light. It doesn't actually deal with anything regarding nervous transmission of signals to the brain. It is there to focus and point that light directly towards the retina, okay? So the posterior segment over here, which actually does contain the retina, which contains those sensitive photoreceptor cells, the posterior segment contains the retina and will receive that incoming focused light so that an electrical signal can be sent to the brain. So you can see the optic nerve back here. That's that nerve pathway that's going to take the information we get from the light striking these retinal cells and take it to the brain so it can be processed. Okay, so let's talk a little bit about the anatomy of the anterior segment. So a couple of structures for us to keep track of here. You have the cornea, which is this outermost cell structure right here. This is actually just a transparent layer of cells that both protects the inner chambers of the eye, so keeps bacteria from getting in, and then it also acts to refract or bend light towards the retina, okay? So when light enters your eyeball, when it strikes the cornea, that light is going to get bent towards the lens, which we'll talk about in a second, and then bent again as it goes through the lens and goes towards the retina. The iris, which you can kind of see right here, this isn't the best angle to really appreciate what the iris looks like. The iris acts kind of like the diaphragm of a camera. It is a muscular structure that can either dilate or constrict the pupil, which is your hole-like opening here that allows light through. So it dilates or constricts the pupil to determine how much light passes to the retina. So your iris is responsive through the autonomic nervous system to how much light you are ambiently being exposed to. When you are in a very bright light environment, your pupil will be very constricted. And when you're in a dark environment, your pupil will be very dilated. This is a autonomic reflex that your brain and your nervous system undertakes to make sure that you are not overexposing your retina to too much light. The lens, which you can see right here, is just as it sounds. It's basically a big globular, clear, refractive body that is suspended in these little zonular fibers here, and it is connected to muscle. And what we will see later is that not only does the lens refract and focus light that is entering into the eyeball, it can actually be changed through the muscular contractions of these ciliary muscles, which can mold and shape the lens to refract and focus the light of moving or distant or near objects. So that's a process we'll talk about a little bit later. So for the posterior end, the retina is at the back of the eye and consists of multiple layers of cells. So it's not just photoreceptor cells, although that's definitely where our focus will be. And this obviously includes those light-sensitive photoreceptor cells, which include both rods and cones. And we will have a substantial discussion on the differences between rods and cones here in just a little bit. Underneath the retina is the choroid, or more appropriately called the choriocapillaris, although we'll just call it the choroid. This is basically the blood supply to the retina. And that's very interesting because you'll hopefully recall from our chapter on hypoxia that the retina is under the highest oxygen tension of any tissue in the human body. Those cells in the retina have a dramatically high need for oxygen, which is why when you enter the indifferent stage of hypoxia at relatively low altitudes, so between 5 and 10,000 feet MSL, the very first symptom you're likely to experience is a little bit of loss of vision at night. It's because those cells and those tissues in the retina are most sensitive to oxygen deprivation, and they are going to experience the loss of function before any other part of your body does first. Okay, let's talk just a little bit about the physical nature of how light refraction works. So this is described by what is called Snell's law. So what this law says is that when light passes between two media of varying refractive indices, the light will be bent or refracted. Well, what do I mean by media? Well, so a medium can just refer to some kind of substance through which light can move. So take this example here. So here in red, we see a ray of light, which is passing from one medium in white into another medium, which is blue. So just for the sake of argument, let's say the white medium is air and the blue medium is water. So when light passes from the air through the surface of water, that light is going to be bent because the air and the water have very different physical properties, and therefore they have different refractive indices. So that light will be bent as it moves from the air and into the water. If you've ever swam in a swimming pool, you've probably noticed this before. If you drop an object down to the bottom of the pool, the location of that object is actually kind of hard to pinpoint because you're seeing basically a visual illusion of that object because you are looking at it from above the water and its actual location below the water is different than how it appears to your eye because the actual location is being offset because of that light refraction. So Snell's law, like I said, describes the relationship between the refractive index of one medium, the angle of incidence, and the angle and the refractive index and angle of refraction of the other medium. So I'm noticing a typo here. That should be theta 1. So that's just the angle at which the light enters the second medium will help us determine what the angle coming out is. So don't worry too much about those variables. We are not going to do any mathematical calculations here. I just want you to understand that when light moves from one medium to another, if the angle is right, if the angle's not 0 or 180 degrees, then that light is going to get bent, and how much that light gets bent is going to depend on the refractive index difference between the two media. So arbitrarily, the refractive index of air is set at 1.00. For us, talking about the eyeball, this is highly relevant because all the light that is going to enter our eye is going to come from the air that's all around us. And then the refractive indices of the cornea, the aqueous humor, the lens, and the vitreous humor, these are all the media that light is going to have to travel through as it makes its way to our retina. These refractive indices are 1.38, 1.33, 1.40, and 1.34 respectively. So what this means is that when light passes from the air into the cornea, there is a difference in refractive indices between those two media. So that means the light is going to be bent. As the light moves from the cornea, which is 1.38, into the aqueous humor, which is 1.33, the light will be bent a little bit more, but it's not going to be bent nearly as much because there is a much smaller difference between 1.38 and 1.33 compared to 1.38 and 1.00, okay? So every time light passes from the air to the cornea to the aqueous humor to the lens to the vitreous humor, that light is going to be course corrected and bent just a little bit. The greatest amount of refraction is going to occur at the cornea and at the lens. You wouldn't think the lens would cause much light refraction just because of the small difference in refractive indices, but the nice thing about the lens is that the shape of the lens actually matters a lot too. So the lens is going to refract light a lot, not really because of its refractive index, but more because of its shape. So we'll talk about that a little bit later. So the idea here is that rays of light which enter into the anterior part of the eye are going to be bent or refracted off of the cornea, the aqueous humor, the lens, the vitreous humor on its way to the retina. And what we are ideally trying to get to happen here is those light rays will all be bent in such a way that the light rays will converge onto a singular focal point right on top of that retina. So if we can get that to occur properly, that's going to be how we get clear, crisp 20/20 vision. So we'll talk a little bit later about, well, what happens if somebody doesn't do that right? What happens if somebody is short-sighted or far-sighted? What happens if somebody has problems with how they refract light? What happens when you don't actually create that perfect focal point right on top of the retina? So we will talk about that at a later point. First, let's talk about the accommodation process. So, we definitely get a lot of refraction off of the cornea, but let's talk a little bit more about the lens for now. So, the amount of refraction you get from the cornea and the humors is basically static. It's set in place. It's based on curvature, it's based on refractive indices, and the curvature of those structures is not really something that's going to change much over time. But the curvature of the lens can be changed very dramatically and very dynamically in a very short amount of time. And this process is called accommodation. So, this change in the curvature of the lens, which is possible because of that muscular structure that suspends the lens in place, this allows us to adjust the focal point that strikes our retina to allow us to accommodate for moving objects or objects that are at varying distances from the eye. So essentially the way this is going to work is that ciliary muscle that suspends the lens in those zonular fibers, the muscle will tug or push on those fibers, and that will allow us to change the sphericity of the lens according to a number of sensory inputs that are coming into the brain. We're not going to get into the nitty-gritty details of how those sensory inputs work, but essentially, your lens can change its shape dynamically over the course of just a few seconds to make sure that when you change your focus from an object that's right in front of you to an object that's very far away, it doesn't take too long for you to get a good, clear grasp of what you are looking at a far ways away. It's because your lens can change its shape to make sure that the focal point of what you're looking at is always right on top of your retina. Okay, so let's switch gears now and talk about the difference between seeing during the day and seeing at night. So the human eye employs two different visual systems within the same retina, and that's going to be what allows us to see during the day and what allows us to see at night. Your day or light adapted vision is called photopic vision, and this is going to be the type of system your visuals rely on when there is a lot of light present. At night, you are going to use your scotopic vision, which is what you use when light is very scarce and you don't really have a whole lot of light to work with. So this is going to be where we start to distinguish between rod and cone photoreceptors. Rod photoreceptors are highly sensitive to light, meaning that they don't really require a whole lot of light to work very well, but they don't really account for any ability to distinguish color. So if you're just relying on your rod photoreceptors, you would just be seeing black and white or shades of gray. So rods are going to be responsible for our scotopic vision. So again, your scotopic vision is going to be your night or dark adapted vision. There's not very much light to work with, so it's a very good thing that rods are so sensitive because they can work with very low amounts of light. Cone photoreceptors are not nearly as sensitive as rod photoreceptors, and thus they do require more light to be present, so they're really only going to work for your photopic vision, but they do allow us to distinguish colors, and they do account for our clearest, crispest images that we are able to see. The interesting thing, though, is that if you did a head count of all the photoreceptors in your retina, rods are going to account for about 99% of them in all parts of the eye except basically the very center of your eye, which is called your fovea. Your fovea contains a very high concentration of cones. You can almost think of the fovea as being like a big island that is made of cones floating in a sea of rods. So your cones essentially are going to account for your central vision. If you look straight ahead and you focus right dead straight ahead of you, you are using your cones to collect the visual information about what is right in front of you, whereas your rods fill in the information on the peripheries. So if we kind of look real quick at the cells of the retina, so if we look right at the very back of the eye here, so here's the anterior segment of the eye up here, here's the posterior segment back here. So light is going to pass through in this direction here, and again, you can see all different types of cells here. The ones we want to focus on are called the rods and the cones, and as you can imagine, the rods look like rods and the cones look like cones. So light is going to come in this direction and stimulate those rod and cone photoreceptors, and then they are actually going to send electrical signals back the other way, and eventually that will make their way to the fibers that actually make up the optic nerve and allow us to transmit information to the brain. Okay, so these other cells here, like bipolar cells and amacrine cells and ganglion cells, don't worry so much about those. I'm not going to require you to really know anything about those. So the main thing you want to know is that light comes in this way, stimulates these photoreceptor cells, and then the signal actually comes back the other way and goes through those optic nerve fibers. So the basics of how this works is that light will activate a pigment that is inside these little hairbrush portions of the photoreceptors that you see here. Okay, see those? So that pigment is a complex of protein and a vitamin A derivative. So those are in the photoreceptors, and this is actually going to lead to a series of chemical and electrical reactions that will then be passed from cell to cell in the retina, which results in an electrical signal traveling through the optic nerve into the brain. Okay, so we had just previously talked about the asymmetrical distribution of rods and cones. So cones are at the very center of your retina in that fovea. So you can see if we do a head count on the number of cones, you don't really see hardly any cones in the peripheral parts of your retina on either side. But once you get to that central fovea, you see a big spike in cones. And then on the peripheries, you have lots and lots of rods, but then you get to the fovea and you have no rods whatsoever. But then you get away from the fovea and you get rods coming back once again. So the fovea, the center of your eye, that is going to be your color vision. That's going to be your crisp, clear color vision because that's where the cones are. Your peripheral vision, that's where all the rods are. That's going to be where you have your much more light-sensitive vision, your peripheral vision, but that's not going to be in nearly as much detail, and it's not going to have much color associated with it because there's not many cones over there. You will notice that there is this one part of the retina for which there are neither cones nor rods. This is what is called the physiological blind spot. This is the portion of the retina for which there are no photoreceptors present whatsoever, no rods and no cones, and that is because this is the location of what is called the optic disc. The optic disc is the anatomical structure in the back of the eye through which the fibers of the optic nerve exit out the back of the eye. So because that is the exit point for basically a big bundle of wires, there cannot possibly be any photoreceptors there, and that is what we call our natural or physiological blind spot. So if we go back here, you can actually see your optic disc right there. There's your fovea right there, but this is your optic disc right there. And then we can see that right there again. There's your fovea and there's your optic disc. So that is your natural physiological blind spot. So here we can see the same kind of thing. So there's your optic disc right there. That's not only where the optic nerve fibers exit out the back of the eye, that's also where blood vessels like arterioles and venules enter and exit the eyeball. And then here you can actually see what's called a fundoscope image of your retina. So this is what an optometrist might see when they actually dilate your eye and look into the back of your eye to look for signs of retinal degeneration or retinal detachment. So you can actually see the optic disc right here. So there are no photoreceptors there. So if you shine light right on that optic disc, you are not going to see it because there are no photoreceptors there to be stimulated. All right, so with all that in mind, now that we have kind of a general sense of the difference between rods and cones, and we are definitely not done talking about those yet, so we'll bring those back later, let's talk about visual acuity a little bit. So the ability for us to distinguish fine detail in our vision, as you can imagine, is very important for flying an airplane. So we need to be able to see visual landmarks. We need to be able to see the controls right in front of us. And then we need to be able to look out for other aircraft that are flying in our vicinity. So this is all going to be especially true for pilots that are relying on visual cues when flying VFR, and it's going to be important for flying IFR too, okay, just for different reasons. So, our greatest visual acuity occurs when an object is in the center of our vision, which again is where your cones predominate. Your cones are not as sensitive as rods. All that means is that cones require more light to operate properly. But if there is an appropriate amount of light, those cones will produce a clearer and crisper and better image than the rods otherwise would. So if we want to see in as great of detail, in as fine of detail as we possibly can, we want to be using our photopic vision, and we want the object that we're looking at to be right in dead center in the middle of our vision. So like I said, cones are less sensitive than rods, which means that they require more light to be present to be effective, but their ability to make out fine details is much better than the rods, which are concentrated in the periphery of our retina. So what you're seeing here in this graph on the y-axis is a measure of visual acuity on a scale of 0 to 1, 1 being the best. You get your best visual acuity right here in your central vision, right at the fovea. You can see your visual acuity, or your ability to make out fine details, diminishes greatly and precipitates greatly the further away you get from the fovea, and that's because you're getting away from using those cones and you're having to rely on rods, which again are more sensitive to light, but they don't produce as fine of detail. So why is it that cones produce better detail than the rods do? Well, there's several reasons for that, but the physiological reason that we're going to focus on here is because of the ratio of rods to the other cells in the retina and the ratio of cones to other cells in the retina. So a single optic nerve pathway is going to receive input from multiple rods. So what you're seeing here, this cell down here is called a retinal ganglion cell, and its axon will actually make up the nerve pathway that leads out through the back of the eyeball through the optic nerve. So what you're seeing here is that if this cell gets excited and sends a message to the brain, it's receiving input from, just based on this picture, 5 different rods. So 1, 2, 3, 4, 5. All 5 of these rods are giving chemical input into this one retinal ganglion cell. So basically, you have to stimulate 5 rods to get one signal to go through this cell. But the ratio for how it works with cones is totally different. This one cone that you see right here stimulates one ganglion cell. This one cone that you see here stimulates one ganglion cell. So you only have to stimulate one cone to get the same signal traveling through those retinal ganglion cells. So what this means is that if you engage all the cones in your retina, in your fovea, then you are going to get more signals traveling towards the brain. And since the brain is receiving a greater amount of information about what you're seeing, the brain is able to piece together that extra additional information and produce a lot more detail. So here you're stimulating 5 cones just to get one little piece of information. So if the brain's not receiving that much information, the brain's not going to really know 100% what to do with that, so it'll produce as little detail as it possibly can. So again, this difference in ratio between photoreceptors and retinal ganglion cells allows a single cone to transmit more detailed information to the brain compared to the rod, which this is part of what helps to explain why our visual acuity in the fovea is much higher than in the periphery. Okay, let's talk about field of view. So the difference between foveal and ambient vision. So our field of view spans about 200 degrees to the side, as you can see from this diagram. So the very limit looks like it's about 208 degrees, 104 here and 104 here. So we'll call it about 200 degrees. And then vertically, it's about 120 degrees, 70 degrees down and 50 degrees up. So clearly, we can see a lot of what is around us, but we can't see everything. So although our peripheral or ambient vision, which again is focused on rod function, This does not have high visual acuity. Yes, that is true, but your peripheral or ambient vision is still very important for your spatial orientation, your own coordination, and your ability to see moving objects, okay? So, as we will discuss later, there is a type of optical illusion where it is hard to see an object moving if it is right in front of you. It's a lot easier to see an object moving if it is in your peripheral vision. So that's something to keep in mind. So foveal vision not only produces the highest visual acuity, but it also allows us to see colors clearly and accurately. But as I just said, one of the limitations of foveal or central vision is that it makes seeing moving objects a little bit hard. Okay, so one of the main reasons why we have gone to all of the pains of going through all this and learning about all of this is because regardless of whether you're flying in clear conditions in VFR or if you're in instrument meteorological conditions where you're having to rely on your instruments to know where you're going, you as a pilot are going to be required to maintain vigilance in seeing, detecting, and avoiding other aircraft. You can't necessarily just rely on information that you're getting from radar or from ATC or any other sorts of technologies aboard on board your aircraft that detect other airplanes and give you a heads up. It's still best practice for you to use your eyes to look out of the cockpit and spot other aircraft and do what you need to do to avoid them. So it turns out that many near mid-air collisions, so a mid-air collision that could have been but was avoided, many of these are prevented from becoming actual collisions because pilots did see the other aircraft in time to make evasive actions. However, most actual mid-air collisions occur because the involved pilots did not see each other in time. So these were cases in which the pilots could have conceivably spotted each other, but for whatever reason did not. So what this indicates, not necessarily that somebody screwed up, but it maybe indicates that there are these severe limitations that are inherent in human visual performance. Like I said, it is not a perfect physiological process, and this suggests more guardrails should be in place to help us prevent mid-air collisions. So let's talk about some of the physiological defects that actually manifest themselves while you are flying your airplane. The first one we'll talk about is called accommodation lag. We talked about the accommodation process involving the reshaping of the lens to adjust its curvature so that we can maintain a good focal point on the retina for a variety of different objects. But the problem here is that if you're dealing with a fast-moving object, that's going to produce lag time. It takes your eyes a couple of seconds to adjust to a new object in your field of view or an object that's moving faster. So it will take your eyes time to adjust to the object to see it properly. And if you're dealing with a very fast moving object, that lag time is greater. And if you're talking about avoiding a mid-air collision, maybe that lag time, a couple of seconds, maybe it makes all the difference in the world. With empty field myopia, this describes a phenomenon that when you are either flying at very high altitudes with no visual landmarks, or it's cloudy and you don't really have anything to look at to distinguish themselves, if you're just kind of staring out into empty space, your eyes as a pilot are going to just naturally settle into their resting state, which is basically to focus on what's right in front of your nose. So this is going to only allow for clear vision at about 2 to 6 feet away. So again, myopia is basically just nearsightedness. So this is a type of nearsightedness that sets in when you don't have distant objects to focus on. So we'll talk about strategies to overcome this a little bit later. So the blind spot we talked about before, so the physiological blind spot that we talked about previously, you really don't ever notice because the blind spot of your left eye is offset by the blind spot of your right eye. They compensate for each other. So as long as your vision in both eyes is unimpeded, the visual field of one eye can compensate and fill in the missing information for the other eye. But if you are dealing with an object impeding the vision in one of your eyes, like maybe a wing or kind of a panel by the side of the window of the cockpit, if you have your vision impeded in one eye, that is actually going to cause the other eye to have information that is not compensated for or not filled in for, and then that other eye's blind spot can potentially become a problem. So with peripheral vision, we saw previously that visual acuity is highest with foveal or central vision. Objects in your peripheral vision that appear stationary will be very hard to see, but the opposite is true for foveal vision. If an object is moving in your central vision, it can be kind of hard to see. So understanding kind of the difference between your peripheral and central vision and what each is good for and what each is not good for is going to be very important for you. Central vision blindness at night. Since night does not provide adequate light to activate the cones in your fovea, you're basically going to be relying on your peripheral or rod-based vision at night. So if you're flying completely in the dark, your cones are not going to be working at all, okay? So remember, the cones are not all that sensitive. They need lots of light to function properly. So basically, if you're flying at night, you are actually going to have two physiological blind spots in each eye, one for the optic disc and one for the fovea. And we will talk about strategies to overcome this a little bit later. And then your book also makes mention of what is called the blossom effect, which describes the phenomenon of an incoming aircraft only becoming big enough to see when it is basically too close to avoid. This is, again, one of those limitations of foveal vision where it is very hard to appreciate a moving object if it is right in the center of your vision. So that object will appear to be stationary up until the point where it is basically it gets really, really big very, very fast, and it is basically too close to avoid at that time. So your book estimates that the reaction time necessary to avoid that aircraft as soon as it appears at the very end of your vision is about 12.5 seconds. And that seems like a long time to make a decision, but first you have to figure out, oh, well, what is that way up there? Is that an incoming aircraft? Is it coming right at us? Is it like going in the same direction as us? What do we need to do? So there's lots of time that has to be dedicated to seeing the object, having that information about what you see be transmitted to your brain, using your brain to process that information, figuring out, okay, well, what do we need to do about this? Do we need to make evasive actions? Sending a message from your brain to the muscles in your arm and in your hand so you can operate the flight controls to take that evasive action. So if you look on Blackboard, I posted a link to an online reaction time test. And the main thing I want you to understand here is that things in the nervous system operate very quickly because we're dealing with electrical signals, but very quickly is not the same thing as instantaneous. The reason why we have reaction times is because physiologically it takes time for information to travel through the body, and it takes time for information to be processed. So 12.5 seconds may seem like a long time, but it really is not very much time. So you're really, really, really working on making sure that you can identify things properly and react quickly as well. So again, this is kind of a illustration showing you what the blossom effect is like. So if you see a distant object in the distance, and basically the time it takes for it to actually get big enough for you to appreciate what it is, it's basically too late at that point. It might have been 10 miles away here, 6 miles away here. So once it gets to be 5 miles an hour, you would think, oh, well, it's going to look a lot bigger. Well, it doesn't really look a lot bigger. So it really only gets big enough to see when it is very, 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 very close. So again, it takes time for this incoming aircraft to be recognized as such, and that's really only just a part of what's necessary to avoid it. The brain has to send messages to and from the muscles, so we have to have lots of different things happening inside the body that all take time. So that is what produces this reaction time of 12.5 seconds. See the object, recognize that it's an aircraft, become aware that it's on collision course with you, make the decision to turn left or right, having the signal sent from your brain to your muscle. So it all takes time. So this is a table from your textbook. I'm not going to go through all of it, but these are just some environmental factors in midair collision avoidance. So conspicuity, so like what does the aircraft look like? Is it like easily detectable with your eyes? Does it have kind of structural features that might make it easy to spot? Glare, so light reflecting off of aircraft from the sun. Reduced visibility conditions, aircraft blind spots. So depending on kind of structural features of your aircraft, such as Cessnas, which always seem to have their wings mounted on the top of the fuselage, versus Pipers that always seem to have it kind of below the level of the fuselage. So this is actually going to produce kind of different effects for what your field of vision looks like while flying your aircraft. So this kind of really underlies the importance of understanding the aircraft that you are flying and what its limitations are. So as we've talked about, and as you can hopefully continue to imagine, there are all sorts of problems that present themselves when you are trying to fly at night. So statistically, it is about 2 times more likely to experience some kind of fatal accident when you are flying at night, if you're just flying VFR. If it's a clear night and you can actually see your landmarks, just because it's night, you're 2 times more likely to have an accident. And if you're flying in instrument meteorological conditions at night, that likelihood goes up to 5 times more likely. So a big reason for why the dark is so dangerous is because of what's called dark adaptation. So you understand what that means. You understand that it takes time for your eyes to adjust to the dark when you've been in the light. So what this has to do with is rod sensitivity. Rod sensitivity depends on the abundance of rhodopsin. This is the photopigment that is in all photoreceptor cells that we talked about before. Rhodopsin, functional rhodopsin, is a combination of a protein called opsin that is produced by these cells, which complexes with vitamin A. And together, this makes the pigment that absorbs photons of light, produces chemical energy that is then used to transmit electrical signals to the brain. So this pigment can be quote unquote bleached from excessive light. If rhodopsin is bleached, then it is temporarily disabled and cannot absorb any more photons of light until after about another 30 to 45 minutes. So if you are going from very bright conditions to very dark conditions, it's going to be very hard to see in those dark conditions because you have so many photoreceptors, so many rods that are currently bleached, and they need about 30 to 40 minutes to recover or reset. So going from light to dark conditions very suddenly and very drastically will result in very greatly diminished vision until after that roughly 30-minute adaptation process has completed itself. Another thing to be aware of when flying in the dark is what is called the Purkinje shift. So rods in general are not stimulated by wavelengths above 600 nanometers. So that 600 to 700 nanometer range of the visible light spectrum is really where you get all of your oranges and reds. So scotopic vision is really going to shift towards the blue-green end of the spectrum. So you're not really going to see or you're not really going to be stimulated with those red colors anymore. You're going to be stimulated a lot more with those blue and greens. So this will actually affect your dark adaptation process. And kind of an old antiquated practice was that pilots would often use red light or red tinted goggles to enhance their dark adaptation. So when they're flying in the light, they'd kind of tint their eyes with red light to make sure that we are only stimulating the rods with those blue and green lights so that when it does get dark, those rods are kind of already adapted to what they're going to have to deal with in the dark. So there's kind of some problems inherent in this. So what's actually recommended nowadays is rather than using red light, it's recommended that cockpits be illuminated with very low density white light, which engages what's called mesopic vision, which is kind of a happy medium between photopic and scotopic vision, and it utilizes rods and cones simultaneously. If you look at the picture over here, and you look at this flower, which is obviously very red, this top image is what you would see with photopic vision. The bottom image is what you would see with scotopic vision. Obviously, all the color detail is gone. But with mesopic vision, you can still see the object and make out a little bit of color, but you're not making out as much color because there's not really enough light to fully engage those cone photoreceptors, but there is enough light to engage your rod photoreceptors to fill in some of the details that you would otherwise be missing. So this practice of using the low-density white light in the cockpit will allow for maximum dark adaptation while still allowing for some color discrimination. But one thing your textbook warns about, and this makes total sense, is that care should be taken when you're flying at night and you go near a thunderstorm. If you get exposed to a lightning flash, that could totally ruin your dark adaptation by suddenly switching you to photopic vision, bleaching those rod photoreceptors, and then it's going to take 30 minutes for you to adapt to the light again. Okay, so we're nearing the end of the chapter. We still have a few things left to cover. Visual hypoxia. We talked obviously in chapter 4 about hypoxia, what oxygen is and why oxygen is necessary for tissues in the body, and how oxygen gets taken into the body and transported all throughout the body. We learned in that chapter that the retina has the greatest oxygen demand of any tissue in the body, so supplemental oxygen is necessary when flying above 5,000 feet MSL if you're not in a pressurized cockpit or cabin. So the reason for this is to prevent visual deficits. We learned in chapter 4 that it's really only above 10,000 or 12,000 feet that you start to experience some of the other kind of more conspicuous effects and symptoms of hypoxia. But the very first thing you'll see as early as 5,000 feet is a loss of night vision. And that's because these cells in the retina have such a high demand for oxygen that you'll notice a decrease in their function before you notice anything else. So this is what's sometimes called visual hypoxia, and it is definitely compounded at night because dark adaptation will take longer to occur if those cells are not getting enough oxygen, and color vision will be diminished as well. Another interesting phenomenon is called corneal hypoxia. So this describes the visual deficit that occurs when the cornea, which again, these are not light-sensitive cells, but they do refract the light that focuses the light on the retina. When these cells do not receive enough oxygen, they start to their function starts to be hampered as well. So those corneal cells do not get their oxygen from the choroid like the retinal cells do. No, these corneal cells get their oxygen from the outside atmosphere. So the cornea is all the way over here on the other side of the planet that is the eyeball. So they don't get their oxygen from the cornea or from the choroid like the retinal cells do. And then finally, carbon monoxide will also hamper oxygen delivery to the retinal cells as well. So that's more of a anemic hypoxia, as we talked about in chapter 4, but it does affect the eye as well. Okay, so let's talk about optic illusions. So one of the major optical illusions we're going to talk about here is called the autokinetic effect or autokinesis. So this is the phenomenon of the appearance of a stationary object or stationary light moving around at night when stared at directly. This is exactly what I talked about when I said that foveal vision has limitations associated with it. And especially at night when your cones aren't really working, it can be very hard to focus on a light that is right in front of you. And basically because the brain is having to kind of fill in information about that object that you would otherwise be getting with your cones, the brain can easily be fooled into thinking that that object is moving around when it is not actually moving around at all. So this again emphasizes the importance of your peripheral or ambient vision in being able to detect accurately when objects are moving around. So one of the best practices is to kind of take a side eye approach when you're looking at objects when flying at night, kind of move your head around and make sure you're getting those into your peripheral vision so that you can get an accurate sense of where those lights are. So as you can imagine, this can make watching for other aircraft at night very tricky. Okay, so color blindness, as you probably were aware, we're going to get into this. So color blindness is important, and this will actually kind of lead into a new detail that we need to learn about. There are 3 different types of cone photoreceptors. There are what are called S cones or blue cones. The S stands for short. There are M cones or green cones. The M stands for medium. And then there are L cones or red cones, and the L stands for long. So each type of cone contains a different kind of rhodopsin pigment that absorbs different wavelengths of light. The blue cones will absorb these wavelengths, the green cones will absorb these wavelengths, and the red cones will absorb these wavelengths. Okay? So that is how we're able to see all the wavelengths in the colored light in the visible light spectrum. It's because we have 3 different types of cones that give us complete coverage from 400 to 700. So you'll recall that the rhodopsin pigment is made of a protein component and a vitamin A component. The protein component is what is different between blue cones, green cones, and red cones. So the instructions, the genetic instructions for making that opsin protein for that part of the pigment are encoded in the DNA. And those instructions, which are called genes, that gene for making that opsin protein is specifically located on the X chromosome. Now, these are called the sex chromosomes. Women have 2 X chromosomes, whereas men have 1 X chromosome and 1 Y chromosome. So the issue with this is that because these cone opsin genes are located on the X chromosome, if you, hold on here. Yeah, so I'll get to that in a second. So just real quick, let's talk about rhodopsin. So this is what rhodopsin is generally going to look like. It embeds itself in the cell membrane of those photoreceptor cells. So you don't have to worry about this too much. Kind of the spirally parts of this are the protein or the opsin. And then that big globular thing in there is the vitamin A that has wedged itself in there. So rhodopsin is the pigment found in rod-encoded photoreceptors that absorb photons of light and convert that energy to the chemical energy needed to send signals to the brain via the optic nerve. And like I said before, this rhodopsin has 2 components: the opsin, which is the protein that is encoded in the DNA, and then you have the retinal or vitamin A that is obtained through the diet, and it will change its structure depending on which stage of the visual cycle it is in. So when we talk about photo-bleaching the photoreceptors, we're talking about causing a change to that retinal structure that takes time to reverse itself. So back to what we were talking about with color blindness. If someone is born with a mutation to one of those cone opsin genes that causes those cone opsins to not be made properly or become non-functional, that can actually lead to specific kinds of color blindness, meaning that you no longer have complete coverage from 400 to 700 nanometers. So men are far more likely to be partially colorblind because they only have 1 X chromosome. If a woman has a mutation to one of their cone opsin genes, it's very likely that they actually have a good copy of that gene on the other X chromosome, which can compensate for it. But men, since we only have 1 X chromosome, if we have a bad cone opsin gene, we don't have another X chromosome to compensate, so we're more than likely going to reap the quote-unquote benefits of being colorblind. So what you're looking at here is what is called an Ishihara color test, which can be used to help to diagnose different types of color blindness, whether it is red-green color blindness, blue-green color blindness, or otherwise. So medical certifications for being able to fly will actually require pilots to be able to perceive color to the extent that they need to be able to safely operate their aircraft. So table 6.2 from your textbook shows you the different types of colors that are used in flight operations. So green, green, red, red, white, green, red. So you can see red and green show up a lot here. So, unfortunately, red-green color blindness is one of the most common types of color blindness, which unfortunately, again, are two of the most common colors used in flight operations. So, this would more than likely exclude anyone with red-green color blindness from being able to fly safely. Real quick, we'll talk about visual acuity testing. You have more than likely seen this type of chart before. It's called a Snellen chart. This is used for trying to determine what degree of visual acuity someone has, with 20/20 being perfect vision. And then as you go up from there, vision gets more difficult, meaning that if you had 20/200 vision, you would have to be 20 feet away from this object to see it, whereas someone with normal vision could be 200 feet away from it and still see it. So obviously that's kind of a big deal with flying. So deficits in visual acuity are usually going to be due to refractive errors. It would be easy to think that, okay, well, if I'm not seeing something properly, it's because of my photoreceptors or because of my retina. Well, that's not actually the case. So it's usually going to be because you are not doing a good enough job refracting light. So that indicates that the problem is not with your retina, but it's more than likely with your cornea. So the cornea, which is made of living cells, the cornea can actually grow and expand and change its curvature over time. And that can lead to an eyeball that is either too curved and refracts light too much, or an eyeball that is too flat and refracts light too little. So this can lead to visual acuity deficits like myopia and hyperopia. Myopia, or nearsightedness, occurs when the cornea is too curved and it refracts light too much, resulting in the focal point being in front of the retina. Okay, so you can see your focal point is right here rather than right here where it's supposed to be. So this is going to cause those light rays to be out of sync or out of phase with each other when they hit the retina, and that's going to produce a blurry image. Similarly, for hyperopia down here, the cornea is too flat and refracts light too little, such that the focal point is actually behind the retina, and you get a fuzzy image or a blurry image because those light rays are out of focus or out of sync with one another. So the way that you fix this is with corrective lenses or contact lenses. So if someone wears glasses, and you will either use a concave lens with nearsightedness or a convex lens with farsightedness, what this extra lens will do is provide an extra layer of refraction, and that lens is crafted so that it corrects your focal point and pushes it back or pushes it forward to exactly where it's supposed to be. And that is the nature of how eyeglasses are meant to work and correct your vision. It is not a retinal problem, it is a refraction problem, it is a cornea problem. So corrective lenses can readjust that focal point so that it is right on top of the retina. Some other visual disorders to be aware of. Astigmatisms are just irregularities on the surface of the cornea or lens that can cause refractive issues. So these cannot be corrected with just simple convex or concave lenses. They can be corrected with lenses. It's just those lenses have to be very specially crafted for the type of astigmatism you have. Presbyopia is just kind of an age-related loss of visual acuity, and this can be attributed to the hardening of the lens with age, and that will diminish its refractive capabilities and basically make it so that you have kind of trouble focusing on things that are right in front of you. So this can be corrected with just reading glasses or bifocals. Night blindness can be caused by all sorts of things like cataracts, which is just a protein buildup in the lens, the use of drugs, or some kind of vitamin A deficiency from the diet. And then one of the last things we're going to talk about here are some strategies for overcoming night vision deficits, which we'll get to in just a second. And then for now, avoiding mid-air collisions. So now that you've learned the basics of visual physiology, hopefully you can appreciate that while the human eye and the human visual system is obviously very amazing, the eyeball is rife with all sorts of limitations and can be very easily tricked and fooled by a number of optical illusions, your brain too. So here are some strategies for overcoming this. Number one, use a slow and methodical visual scanning technique. So, slowly just kind of scan across your visual field. What you want to remember here is that accommodation lag that we talked about. Give your eyes time to adjust to your foveal vision. Make sure that you're taking time to slowly scan, maybe kind of treat it like different parts of the sky as different quadrants. Look here, okay, look here, focus, look here, focus. So, make sure that you are allowing for that accommodation lag to not become an issue. Consider your aircraft and its blind spots. So your textbook talks about the concept of clearing the area to kind of maybe adjust the your bank angle just a little bit to kind of move those wings around just to make sure that you can kind of see everything and kind of clear some space for your aircraft to make sure that you're getting a clear view of everything that is around you. You want to try to do what you can to avoid empty field myopia, which again, this is the type of myopia that you get when you don't really have anything to look at. So it's suggested that every 2 minutes you focus your eyes on the farthest object possible. If it's clear outside, you can focus on an object on the ground. If it's not clear outside and you're flying an IMC, you can focus maybe on the wingtip or something like that. And then for dealing with the brightness of light, it's suggested that you can wear sunglasses, but you don't want to wear polarized sunglasses because it's useful to be able to see glare off of incoming aircraft. And if you're wearing polarized lenses, that can be a lot harder to see. And then some strategies for dealing with night vision. Maintain dark adaptation. We talked about that before, the use of low intensity white lights. Rely on your ambient vision. We talked about this a little bit. So you want to remember that in the dark, your foveal vision is going to be pretty useless. So identification of objects in flight will have to be done with your rod-focused vision. So it's important to kind of move your head around and side-eye everything to make sure that you're able to use that peripheral vision to see everything. Flying above minimum IFR altitudes, as low-flying objects will be difficult to see. And then be aware of terrain. This goes without saying, but be aware of terrain. Use all resources available outside of visual aids to always be aware of your surroundings. All right, well, that's going to do it for this chapter. I hope you learned a lot. I thank you for your attention, and I will see you next time.